Stereospecificity is the property of being able to discriminate between stereoisomers. And there are many examples in biological systems of stereospecificity. It's the way pheromones work, it's the way enzymes work, our olfactory receptors, taste buds, etc. You've got here, for example, two compounds that are enantiomers, a carbon plus and minus carbon, in which the chiral carbon is this one here, and they taste different. So that's just an example of how a molecule with one chiral carbon is already detected differently by biological systems. There are many examples of stereospecificity in biological systems. It's the way our antibodies work, the way our enzymes work. Proteins, stereospecificity in proteins is hugely important, and the way receptors transmit information within the cell or in between the cells is based strongly in this stereospecificity, in this ability to be able to detect distinguish between stereoisomers. Stereoisomers and stereospecificity is also hugely important in the pharmaceutical industry, and in many cases a particular pharmaceutical drug, only one of the stereoisomers will be the one with the biological and drug activity, and the other one is either inert in the best case scenario or can even be harmful. And a typical example, a very common example, is the case of ibuprofen. The ibuprofen has a chiral carbon, this one here, so it can have, there are two enantiomers of ibuprofen, and we call them R and S. It's beyond the scope of this module to explain what the R and the S means, but I will put some links on Blackboard if you're interested in what R and S means. And only the S enantiomers, only one of the enantiomers is the active one. The other one doesn't seem to have any side effect, and actually there is an enzyme that is able to convert the non-useful isomers into the R isomer into the S one, which is the one that is biologically active. So when ibuprofen is sold, it's sold as what we call a racemic mixture, which means a racemic mixture is a 50-50 mixture between two enantiomers. So in many cases the enantiomer one of the enantiomers is not active. In some cases one of the enantiomers can actually be harmful, and there are some really sad examples of the influence of particular enantiomers, the bad effects of particular enantiomers. And you might be familiar with the case of a pharmaceutical drug called thalidomide. And again, I will put some links on Blackboard to that compound for those of you that want to know more.